Hi everyone. So today's lecture is going to be on circulation and respiration. So let's get started. What you can see here is an illustration. Well, it's actually the real vasculature system, and what they can do is inject it with plasticine, a plastic, so that you can see all of the vessels from the major vessels going all the way down to the very small capillary beds where each cell is able to contact the capillary vessel and make the gas exchange. So it's pretty cool and very organized in its branching. So let's talk about the functions of the circulatory system. Number one, as you know, transport, right? The circulatory system is transporting oxygen, waste products, the immune cells, our white blood cells, our red blood cells, hormones. It is the highway system for the body. But the circulatory system is also used for temperature regulation. So here we have a rabbit that lives in the desert, like a jackrabbit, and you can see that it has very large ears that are very, very vascular. And in this way, the circulatory system can help to maintain body temperature within the optimum range for metabolic function. Remember, we talked about homeostasis, and we talked about the negative feedback regulation that's there. And so what can happen is these vessels here can actually dilate so that more blood is sent to that area, and then that way through convection, remember this is all the last lecture, this is all your terminology from last lecture, then the heat is able to be dissipated. Okay. But the circulatory system also functions in protection. So it contains a variety of cells and chemicals that contribute to the individual's defenses against infection by pathogens, and that is the white blood cells that are in the circulatory system. But also, when we get to the immune system, we talk more about that, you're going to learn that it's more than just white blood cells. Of course, as you know, throughout all of this COVID pandemic, there are antibodies there as well. And then we have what's called the innate immune system that is in the bloodstream as well. So, and we'll talk about that when we get to the immune system, but the circulatory system is very important, right? But just because the circulatory system is very important for us, it doesn't mean that circulatory systems are necessary for all organisms, right? We have to remember that we're talking about the biology of the majority of organisms on the planet as well. And so there are some animals, such as jellyfish, other cnidarians, that don't have a circulatory system. Instead, they obtain their oxygen and their nutrients and eliminate their waste through diffusion. Remember diffusion? So you should be starting to put all of these puzzle pieces of information together. It all should start to become very familiar as I add another piece to your knowledge base. So let's see how that happens. So here we're looking at basically a jellyfish, and we can see here, if you follow my cursor, we've got the what's called gastrovascular cavity, and the mouth is here. This is the external region, and you can see that we've got the water from the outside, the external environment, coming into this gastrovascular cavity. And then as we kind of follow the fluid flow, we can see that we've got nutrient and oxygen-rich fluid connotated in this color here coming down, if you follow my cursor. So oxygen and the nutrients are diffusing into the cells, and then you've got waste products diffusing out of the cells going in the opposite direction. So in this way, the cells have easy access to oxygen and nutrients via diffusion because each cell is located close to the external surface of the body or the gastrovascular cavity. Okay. But when we look at another type of circulatory system. These are what's called open circulatory systems, and we'll see this in most insects. So there's really no clear distinction between the circulating fluid and the interstitial fluid, the fluid that's between the different cells. And so the heart, or hearts plural, a lot of insects have multiple hearts, as you can see here, not just one. We have one, but not every organism has one. So here we can see the multiple hearts in this insect, and they're pumping the fluid mixture, which is called hemolymph, throughout the extracellular spaces inside the body. And so we see this type of circulatory system, this open circulatory system, in insects and most mollusks. Think your snails, right? Is a mollusk. But when we look at mammals, we're looking at what's called a closed circulatory system, and that's what we have. And so blood is contained within vessels that separate it from the interstitial fluid, from the fluid between the cells. And so the muscular heart, there is one, propels blood through vessels to tissues throughout the body. And this is the type of circulatory system, this closed circulatory system, that occurs in all vertebrates. Okay? So the question may be, well, if it's a closed circulatory system and the blood's contained within the vessels and those are separated from the interstitial fluid, how is that exchange happening? Well, let's take a look at that. So first we've got to start with some terminology. So arteries. Arteries are vessels that carry blood away from the heart and to the capillaries. What are capillaries? Capillaries are these tiny porous vessels that bring blood close to the tissue, the cells, enabling the diffusion of gases, nutrients, and other molecules into and out of the tissue. So we can see the artery coming in here, bringing the oxygenated blood. We have cells within this little meshwork that are sitting here that are being supplied by the capillary bed that they're sitting in. And then we have this diffusion that's happening both into and out of the cells. And so the waste products, or the CO2, for example, ends up flowing back through the blood into the veins. And veins are vessels that carry blood away from the capillaries and back towards the heart. And so now the blood is heading back to the heart. Okay. So let's take a look at the closed circulatory system of fish. So fish are a little bit different. They have a two-chambered heart. We have a four-chambered heart. They have a two-chambered heart with a single circuit of flow. So let's kind of follow the flow here. So here we have the atrium, which think of it as the top where the blood's coming in, first chamber, and then you typically have valves in between the chambers. So you have a valve in between the atrium and the ventricle, which sits below the atrium. And so the blood comes into the atrium, the valve opens, and then the blood goes into the ventricle, and then it is pumped along. So let's follow the flow. So here we have the atrium. We have, because of this color coding, you can see that this is the deoxygenated blood returning to the heart, comes into the atrium first, flows into the ventricle, comes through, goes to the gills where it picks up oxygen, right? And now the blood is oxygen-rich blood, and it flows back through the body tissues. All the cells are supplied with the oxygen, give up their CO2, and it flows back. The blood flows back into the atrium, and the circuit continues. So this is a closed circuit with a two-chambered heart, and we see that in fish. Now, let's take a look at the closed circulatory system of mammals and birds. This is what we have, right? So we have a four-chambered heart, and that means there's two circuits of flow. So let's follow the flow. So we have deoxygenated blood that is coming into the right atrium, and we pass a valve here, and the blood flows into the right ventricle, and there's another valve, And the valves, basically just think of it as plumbing. It keeps things flowing in the proper direction, right? If you didn't have a valve, when there's a pump, then blood could go in either direction. You don't want that. You want it to go in the direction that you want that circuit to flow in, and so the valve will close, so the blood is going in a one-way direction, okay? And throughout the circulatory system, there are valves, so it continues to flow in the correct direction. So again, we'll start off in the right atrium, we'll pass a valve here, we go into the right ventricle, and the heart pumps, we pass another valve, and we go to the lungs. Now the blood is deoxygenated coming into the right side of the heart, and it flows to the lungs. It picks up oxygen, right, through diffusion, and we're going to take a closer look at how that happens. So now the blood is oxygenated. See these capillary beds? And we come back into the left side of the heart with our oxygenated blood. We come into the left atrium. We pass a valve. We go into the left ventricle. We pass another valve. The heart pumps, and the blood is pumped out to the rest of the body, where the oxygen is diffused into the tissues. And again, the CO2 is returned throughout these capillary beds, and it returns again to the right atrium. And so it's a whole circuit. It's a four-chambered circuit that happens. So we have the pulmonary circuit, where the blood is pumped to the lungs, where it picks up oxygen, when we talk about the two circuits, right? And then we have the systemic circuit, where the blood is pumped to the tissues of the body, where it delivers the oxygen. So to recap, four-chambered heart, basically two circuits, a pulmonary circuit and a systemic circuit. Deoxygenated blood comes to the right side of the heart, goes to the lungs, gets oxygenated, returns to the left side of the heart, gets pumped out to the rest of the body. Okay? So it's actually quite simple. So let's take a closer look at the closed circulatory system of amphibians. Now they have a different morphology to their heart. They have two atrium and one ventricle. It's a three-chambered heart. So they have two circuits of flow. So let's kind of follow the flow here. So we start off in the right atrium, where the blood is deoxygenated, goes into this co-ventricle, this one ventricle. But the blood basically is sort of staying on its side, right? It's not admixing to the other side. So we can see that the deoxygenated blood that's color-coded here in blue is being pumped with that one pump of the ventricle into the vasculature here, headed off to the lungs to pick up oxygen, returns from the capillary bed in the lungs, and it comes back into the left atrium, goes back into that same ventricle, and gets pumped out to the body. And again, we have capillary beds where the exchange is happening. The oxygen is being delivered, the CO2 is being given back, and we have the deoxygenated blood coming back into the heart. And so this is a very different system, where you have two atrium and one ventricle. Whereas, remember, we have a four-chambered heart, two atrium, two ventricle. Not all organisms have the same type of heart. And you're going to learn more about our heart when you do the lab, where you actually are going to be diagnosing a patient. So that should be a lot of fun. You're going to be using the stethoscope and learning what is being heard with the stethoscope, what that means. I'm going to go into it a little bit here, but it's going to be helpful when you actually hear the sounds during the lab as well. You're going to be doing a cardiac ultrasound, so you can take a look at what that is, what it does, what it's measuring. So you'll be learning a lot, and these are diagnostic tests that either you or someone you know in your family or close to you eventually will be having. So it's good to understand what these things are, these diagnostic tests are. So here it is. This is basically in color, and I'm going to drop this link into this week's lecture for you, so you can go through and kind of play with the different hearts and look at the circulation that they have in the different organisms. And so it's very interactive. It's just a lot of fun to kind of play with. I'll have that link inside the lecture for you. But let's carry on. And we're going to take a closer look at the human circulatory system. That is us, right? So the human circulatory system is composed of a fist-sized heart. So if you were curious about what size your heart is, just make a fist, put it right over your chest. That's about the size of your heart. And it has this intricate system of blood vessels, but as we saw, it has organization, right? We're going from arteries to capillary beds to veins, right? And this is all so that we can transport respiratory gases, nutrients, and waste products throughout the body. So this is another view of the heart sort of sliced in half. And now let's sort of start adding some names to vessels so that you can understand where they are and what they do. So here we have the right atrium, all right? This is where the deoxygenated blood comes into the heart. And the deoxygenated blood comes into the heart through two major vessels. One is the superior vena cava, which is blood coming from the top of the body, right? And we have the inferior vena cava. And this is blood that's coming from the lower half of the body. But they're both opening and dumping in to the right atrium. So the deoxygenated blood is coming from both the upper body and the lower body through the superior and the inferior vena cava into the right atrium. Now, we pass a valve and we go into the right ventricle. Then we pass another valve and we head off to the lungs. All right, we pick up oxygen and then we come back into the left atrium, pass a valve, we go into the left ventricle, and follow my cursor, and then we are going into the aorta. From the left ventricle, we get pumped into the aorta. So you can see that we have some crossing over. Remember, this is in 3 dimensions. It's a 2-dimensional picture of a 3-dimensional object. So the left ventricle will pump into the aorta. And from the aorta, we go throughout the body. We go to the upper half of the body. We go to the lower half of the body. And this is all oxygenated blood. Okay? So this is basically, taking another look at that same circuit that I just walked through, except now we're going to add the capillary beds so we can get a closer look at, where the exchange is happening. And then also we're going to add the names of a few more vessels. Okay, so we're going to take a closer look, except now we're going to take a look at the capillary beds, where the blood is actually going from the vasculature that we just talked about. We're going to add the names of some more vessels, but it's the same circuit. So here we go. Let's start off. So here we have the right atrium. This is where the deoxygenated blood is coming in from, right? So it's coming from the superior vena cava, which is bringing blood from the capillaries of the head and the arms, the upper body, right? And so that deoxygenated blood is going to flow into the superior vena cava, and it's going to go into the right atrium. But remember, we also have deoxygenated blood that's coming in from the capillaries of the lower half of the body, so the trunk and the legs, everything that is below. And so the deoxygenated blood is coming from there and going up through the inferior vena cava into the right atrium. So deoxygenated blood into the right atrium from the body and goes past a valve into the right ventricle, and it gets pumped out into the lungs, and it gets to the lungs by way of the right pulmonary artery, because it's going to, you have lungs on the right side and lungs on the left, right? So the right pulmonary artery is going to the pulmonary capillary beds on the right side, and we have the left pulmonary artery going to the capillary beds on the left side, right? The blood is picking up oxygen, and it's returning back into the left pulmonary vein, returning to the heart. Remember, veins return to the heart. So the left pulmonary vein, it has picked up oxygen and also returning on the right from the lungs through the right pulmonary vein, and it's going into the left atrium because the left atrium is where oxygenated blood returns to, right? So oxygenated blood returning from the lungs through the pulmonary veins to the left atrium. We pass the valve here, we go to the left ventricle, and then it gets pumped out into the aorta. And then from there, it goes back to the capillaries of the upper body and also to the capillaries of the lower body, and the circuit basically starts all over, right? So quite easy. It just makes more sense when you trace it yourself. Sometimes when you take a look at a chart like this, it can look very confusing until you just start to trace it. Because remember I said that in medicine, it's all about logic. So you just follow the flow, and it's all very logical, right? All right. So the heart sounds. When you go to the doctor's office, he puts a stethoscope on you. He's listening to your heart. What is he or she listening for? So the sounds of a heartbeat, this lub-dub, 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 actually come from the two sets of valves that keep the blood flowing in the proper direction. And surprisingly, those sounds are not coming from the heart's musculature contraction. This is what a lot of people think. It's coming from the contraction of the muscles. It's not. It's coming from the valves actually closing. Okay. Sorry about the alarms. That's me making dinner, trying not to burn it. Okay, back to the heart. So the sound of the heartbeat. And so you have the lub-dub. And so the lub is coming from these atrioventricular valves, the valves that are between the atria and the ventricles, hence the name atrioventricular valves. And so you've got one here on the right and you've got one on the left. And so the lub of the lub-dub is coming from the sound of these valves slamming shut, preventing blood from flowing back into the atria so that the blood goes from the atria to the ventricle. Now the dub is the sound of the valves called the semilunar valves, and you've got one here heading off to the lungs, and you've got one here heading off to the aorta, right? And so the semilunar valves slam shut, preventing blood from flowing back into the ventricles, and you can see them here as well when we look at the heart. And so that's what the sound of the lub-dub is. And so it's like doors slamming shut. The heart valves make noises that you can hear with your ear pressed to someone's chest. And you're going to hear that in real time in your lab today. But also there's a link here that you can follow where you can listen to different heart sounds. And so what we're listening for is sometimes these valves do not actually slam water tight, right? Which is what they're supposed to do when they close so that there is no flow in the backwards direction. But sometimes you can get a valve that is damaged, and so it will have, a leak. And so what you can hear is as the valve closes because of that damage, when the heart pumps, you hear a shh sound. And that's exactly what it sounds like. So instead of lub-dub, you hear a ch-dub, ch-dub, ch-dub. So that's because blood is actually flowing past the valves, and that's what you'll hear if you have it happening with the AV valve, and of course it'll happen on the dub side if the actual leak is happening in the semilunar valves, right? So that's what we're listening to with the stethoscope, but also we're listening for the rhythm of the heart, okay? And now let's go into that. And so in the heart, regular and rhythmic contractions are initiated by a region of the heart called the sinoatrial node, the SA node. And this can be measured by using an electrocardiogram or an EKG. We can actually see the electrical activity of the heart. And let's take a look at what that looks like. an EKG reading. And sometimes some people will call it an ECG reading as well. So you may hear both terms. So the SA node, the sinoatrial node, is sitting right here in a region of the right atria of the heart. And so when it fires and the contraction spreads to both atria, as you can see, so the electrical activity fires, both atria fire, and the wave of contraction passes down the center of the heart, bounces back up, causing the ventricles to contract, right? Then the ventricles relax and the process begins again. So it all starts with this SA node. This is also known as the pacemaker of the heart. So when you hear that people have to have their pacemaker replaced, this is the region of the heart that they actually have to replace because it is misfiring. And usually it's a little piece of electronic device that is implanted that will take the place of the SA node to start that electrical activity. And so when we look at the EKG, this is what we're looking at. So each part of this wave has a name. So this very first fire is called the P wave, and it goes P, Q, R, S, T. So P, Q, R, S, and this little hump here is the T. So first fire of the SA node causes the P wave. Then we have the fire here that's going down through the ventricles, which causes the QRS. And then the T wave is actually the ventricles relaxing and the repolarization, getting ready to fire again. And so as you can see, the pacemaker of the heart, the SA node, is responsible for initiating the contractions that pump the blood throughout the body. And it is extremely important, right? If your SA node is misfiring, you're going to have basically what's called arrhythmias, which means the heart is not able to beat with a rhythm. Atria contracting, ventricle contracting, atria contracting, ventricle contracting. And so you have to have that filling, and you have to have the pumping out to the lungs and the pumping out to the rest of the body. And so when you don't have that happening in the proper sequence, this is a problem, and it can be life-threatening. And so this is a closer look at some EKGs. There are lots of things that can go wrong. Here we have what's called a normal sinus rhythm. This is the normal rhythm of the heart. You can see our P, Q, R, S, and our nice T wave. Okay, so we saw our PQRS and our nice T wave after, right? And this is what we're looking for when we're looking at an EKG. We're looking for this normal sinus rhythm. We're looking for P, Q, R, S, T, followed by a P, Q, R, S, T, right? But when we see something like this, this is what's called ventricular tachycardia. And that just means that the ventricles are beating out of sync very fast. And that's what this looks like. That's not conducive to good blood flow throughout the body. That's a problem, right? Here we have what's called a single VPC, PVC, premature ventricular contraction. Sorry about that. So we have a PQRS, a T wave, and suddenly we have this big, huge spike here, which is our PVC, premature ventricular contraction. So what does that mean? That means that we have our normal atria and our ventricle that are contracting, and then suddenly the ventricle just decides to contract again. And that's what you see. That's what you see, this big spike here in the ECG. And then suddenly we have conversion back to a normal PQRST. Now, it's okay to have an occasional PVC, premature ventricular contraction. You can sometimes get these when we drink coffee and you feel your heart kind of do a little twitter. That's because you may be throwing an occasional PVC secondary to the caffeine, depending on how sensitive you are or how much you've had. But if you get into runs of these PVCs, and this is what we worry about, you can get into what's called ventricular tachycardia that can be life-threatening, right? So that's what we worry about. This is atrial fibrillation. You'll see this on lots of commercials for drugs and medications. AFib is what it's called. And this is when the atria are just quivering like this. They're not, waiting, filling, contracting, sending blood to the ventricle as they should. They're just doing this and they're quivering. And so, as you can imagine, that's a problem because, number one, blood's not going to move into the ventricle as it should. When those valves open, there's no contraction to move the blood through. The atria are just doing this. And so if the blood is just doing this, right, inside of the atria, well, that kind of sets up for blood to clot inside the atria. And so with atrial fibrillation, the ventricles are still doing their job, but the atria is not doing its job, and you can end up with lots of clotting problems, which can lead to stroke, etc. This could be life-threatening, and this is what it looks like on an EKG. All right. So, and there's plenty of more arrhythmias. There's a whole study of electrical abnormalities with the heart in the study of cardiology. So it's really fascinating, and you can look into it more if you choose to. So good stuff to know. But let's take a quick look at the structure of blood vessels. Here we've got the arteries carrying the blood away from the heart, right, towards capillaries. Blood at a much higher pressure than in the veins because it's going away from the heart. And so we've got, when we look at the architecture of the arteries, we've got the endothelium, then we have this layer of smooth muscle, and then we have the connective tissue on the outside. And then we get to the capillaries, which just has a single layer of endothelial cells, because remember, this is where our diffusion is taking place between, just in your mind, put happy little cells here, making that exchange of oxygen and CO2 through diffusion in between this capillary bed here. And then we get to vessels. Vessels are carrying blood back to the heart, so we've got much lower pressure in the veins than we have in the arteries. So not as much smooth muscle. There is some, but not as much. And so they are heading back to the heart. But remember, here in the arteries, because we have, that huge push of blood coming out of the heart, the arteries have to deal with a lot of pressure, right? And that's why they have this much thicker and this layer of smooth muscle. But it's this smooth muscle also in the arteries and the veins that are helping to control our blood pressure, because it can contract to make the vessels much smaller, which is going to increase the blood pressure. Just think of water through a hose, right? If I have a big, huge water hose and I've got a certain volume of water going through, if the diameter is really large in the hose, then, it's just going to kind of lazy river through. But if I really make that hose small and that same volume of water, it's going to shoot through. That pressure is going to go up, right? And so the smooth muscle that's in the vessels, as the smooth muscle contracts and makes our vessels smaller, then our blood pressure is going to go up. As they relax, the vessels dilate, the blood pressure goes down, okay? And so this is an actual look at the exchange that's happening at the capillary beds. So each capillary has a diameter so narrow that the blood cells must pass through single file as they go through to make this exchange that's happening in the capillary beds, okay? And this is a look at it. But we also have, this direction, as we already talked about, of the blood flow, right? And these precapillary sphincters, we have these valves that are there. And so these precapillary sphincters can reduce blood flow to your hands or to your feet in cold weather, saving the blood's warmth for vital organs. So your body starts to prioritize after a certain point. When you're out in really cold weather, And the body says, well, what's more important? Because we have to maintain homeostasis, we have to maintain our thermoregulation, and we're now going to start to shunt blood away from the extremities, which you can lose your extremities and still live, in order to keep the core warm, to keep the brain and the vital organs warm. And so the body will actually use these precapillary sphincters and begin to close them off. And so that can be very dangerous if you like to keep your hands and your feet, right? This is called severe frostbite. And this is how people lose their hands and feet in extremely cold weather, because the body will shut off these precapillary sphincters so that blood cannot flow to the extremities in order to keep the core and the vital organs warm. So pay attention when that starts to happen, when your extremities are very cold, when you're outside. Make sure you get warmth. But have you ever wondered why, when you're on an airplane and it's a long flight? I know it's been a while since we've all been on a plane, but you have this long flight. Say it's a 12-hour flight, and all of a sudden at about hour 3 to 4, you'll see every old person on the plane jumping up and walking up and down the aisles. Why is that? Right? Here's why. When you look at your lower extremities, your legs, your feet, right? The blood has to get from your legs and your feet back up to your heart, and there is no other heart to pump it up there. Have you ever thought about that? How does that blood get back up to your heart from your legs and your feet? Well, it turns out that the body is actually using the muscles of the legs to move the blood in that forward direction towards the heart. So here you can see in the illustration, we have the vein. This is the deoxygenated blood. Here we've got the muscles. Say these are the muscles of your calves that are there. And as you're moving your calf, as you're moving your leg, you're actually causing the blood in the vein to move up towards your heart because of these one-way valves. And so muscle contractions help direct the blood flow, and blocking the blood flow from the capillaries or pushing blood through the veins back towards the heart. And so, when you're sitting for a long period of time and you're not using your calf muscles, you're not using your legs, what's happening? Blood's pooling in your veins. That's what's happening, right? And that can be quite dangerous. That can lead to blood clots. And that's why you must get up, move around, walk around, get that blood flowing back up to your heart so that it's not just pooling in the veins of your legs. So eventually, when you do get up after a 12-hour flight, you throw all kinds of clots and you end up heading to the hospital with pulmonary emboli. That's very bad. That's what you don't want. So that's why you see people getting up and walking along those aisles after several hours on a long flight, and you should too, right? And that's also why you should not sit around all day in one spot, that you need to move your legs, get these muscles going. All right, now let's take a look at the constituents of blood. What is in blood? So here we go. If we were to put a small sample of blood in a test tube, which happens every day when you get a blood draw, and then spin it rapidly in a centrifuge, this makes it possible to identify the primary constituents of blood. And basically, we've got plasma and packed cells. So what is plasma? Plasma is the liquid portion of blood. Essentially, it's like a salty water type of solution. It has various molecules, including metabolites. It's got waste, it's got salts, it's got ions, it's got plasma proteins, it's got antibodies in it. All of that is here in the plasma. And then we have the cellular portion of the blood. We've got the red blood cells, which generally make up about 90% of the cellular layer. And then we have what's called the buffy coat. And it's called the buffy coat because it looks like a white layer when you spin blood down. And this is where the white blood cells are. And then we've got, some platelets that are there. And these are what help us to clot, right? So we've got the plasma. If you spin blood down, we've got the plasma on top. You get the buffy coat layer, which has your white blood cells, and then we've got a small layer of the platelets that are there, and then we've got the red blood cells, and that is blood. So when we look at red blood cells, the scientific name is erythrocyte. Erythrocyte, red cell. They transport oxygen from the lungs to the rest of the body. They basically are shaped like this flexible disc. They contain very few organelles, and they're packed with hemoglobin. And this is how they actually transport oxygen. We're going to look at that closer in just a minute. They have this sort of concave disc shape, and they're round. That's really, really important. And so when we look at white blood cells, their scientific name is leukocytes, right? They destroy pathogens, foreign organisms in the bloodstream, and the interstitial fluid. And there's several types of white blood cells, and they differ in their methods of fighting disease and responding to foreign materials. It's kind of like a little army. They each have their own speciality that they do. And so here you're looking at an image of several red blood cells here. This is colorized. Here we have white blood cells, two different lineages of white blood cells. We've got some pathogens here that are being attacked. So platelets, these are sort of the heroes of the body. They're very small. As you can see, here's a red blood cell for comparison. Here's a very small platelet. These are all fibrin threads making up a clot. And so platelets actually help us clot. They slow blood loss by initiating the constriction of blood vessels and the formation of a clot. They can help to release some histamine to kind of get things going. They're composed of small pieces of cytoplasm. They contain no organelles, and they are extremely important because if you didn't clot, that means one little paper cut, you would bleed to death. And so platelets are very, very important. Let's take a look at blood pressure, what that is, and what's being measured. So remember I talked about we've got the smooth muscle in the vessels, and it can contract down and increase our blood pressure, or we can dilate the vasculature and decrease the blood pressure. Well, we can measure that blood pressure and see what's going on. So when we're measuring blood pressure, we get two readings. We get what's called the systolic pressure. This is the force that blood exerts on the artery walls when the heart contracts and pumps blood into the arteries. Okay, so that is the systolic pressure. That is the pressure of the pump, right? And the normal range can be anywhere from 90 to 140, and this is measured in what's called millimeters of mercury. All right, and then the diastolic pressure, which is the lower number here, is the force that the blood exerts on the artery walls while the heart is between beats, so in between that relaxation. So we've got a pump, and then a relaxation, and then a pump, and then a relaxation. And so the pump is the systolic pressure, and the relaxation pressure is the diastolic pressure. Now, the diastolic pressure, normal range is anywhere between 60 to about 90 millimeters of mercury. And so here we can see the readings here on this blood pressure monitor. And so we can see the systolic pressure here is reading about 118, and the diastolic pressure is reading about 84, and it's giving us the heartbeat here beating at 80 beats per minute. That's what that PM is, per minute, right? And then also it's giving you a time. Cool. All right. And so you'll see that when they put the cuff on your arm when you go to the doctor's office, this is what they're measuring. They're measuring your blood pressure. And so there is a chart, and this was recently actually upgraded. The guidelines were changed a little bit. So here we're looking at what normal is. Systolic pressure, we want to see less than 120. Diastolic pressure, less than 80. Pre-hypertension, high blood pressure is hypertension. Remember your Greek and Latin prefixes. Pre-hypertension is a systolic anywhere between 120 and 139, and a diastolic between 80 and 89. Stage 1 hypertension is a systolic between 140 and 159, diastolic between 90 and 99. And stage 2 hypertension is greater, systolic greater than 160, and a diastolic greater than 100. That's a lot, right? Greater than 160. So with a blood pressure that is in a systolic range above 120/80, your heart has to work harder at all times. Remember to get through that really tight vasculature, right? That small diameter. It's pushing against that pressure and it's making your heart work harder with every beat. And then your arteries can start to lose some of their elasticity, right? And all of this can increase your health risk. But also, what can happen is when we get to a systolic pressure approaching 200, that's a medical emergency. That is, get you to the hospital right now. Terrible things are about to happen. You're about to die or blow the retinas off the back of your eye. And so you can get retinal detachment with an extremely high blood pressure. And this is exactly what that looks like. The pressure is so high that the retina here at the back of the eye, which is what we use to see, and we're going to look at that closer when we get to vision in the eye, the vasculature sitting behind it, because the pressure is so high, if it's 200 millimeters of mercury or above, it can just blow the retina and detach it right off the eye. And so what this person, when that happens, this person just, it feels, what they see is what they describe as like a veil, came over their vision. And that's because the retina detached. And that can lead to blindness. And so what they do is, number one, get the blood pressure down, right? And there are drugs and medications to do that. And then they'll take a laser and tack the retina back onto the back of the eye so that the person can retain their vision and then control that blood pressure. So you do not want to sit on uncontrolled hypertension, right? So extremely important. All right, let's carry on. We're almost done. So to pump blood up the steep slope to their head, a giraffe's blood pressure is more than twice as high as in humans and other mammals. And that is their normal. So we have to remember once again that what is normal for us doesn't necessarily mean that it's normal for other species, that other species are going to have their own regulatory set points, right? All right. So let's take a closer look at what a heart attack is, because you'll hear this a lot. Someone had a heart attack. What does that mean? All right. So a heart attack is caused when there is an interruption in the flow of blood through one of the coronary arteries. So, I told you how blood gets to the rest of the body, right? How it gets to the upper body, how it gets to the lower body. It gets oxygenated through the lungs, it gets pumped out to the rest of the body. Well, there are little branches that come off of each of the vessels. Here we can see the branch that's coming off of the aorta, the oxygenated blood, that's going to feed the heart muscle itself, because it is a muscle. It has to have its own vasculature, and those are called the coronary arteries, and they sit right here on the heart, and they branch out to feed the heart muscle. Well, if you get a clot, or if you get what's called atherosclerosis of the coronary vessel, this is a closing of the coronary vessel, and sometimes it can be these calcium deposits, these plaques that begin to harden of the vasculature. We can see that it's causing a narrowing of the coronary vasculature. Well, when it completely narrows down so that blood can't get to it, this region of the heart is going to die, right? Because it needs blood. It needs oxygen in order for their cells to do cellular respiration and metabolism and to survive. And so that is a heart attack, when the vessels of the heart, the coronary vessels, get blocked either by a clot, a blood clot, flying through and wedging in the coronary vessel, or this atherosclerosis happening, that is the closing off, the slow closing off of the vasculature of the coronary vessels, forming these plaques, right? So calcium plaques, cholesterol plaques, it's damaged. And you can see another look as we take a slice through one of those vessels, and we can see this is the section where the blood is flowing through, right? This is the vessel wall here. This is the section that remains for blood to flow through, and this is the huge fatty plaque that's clogging the coronary artery, and that's going to damage the muscle of the heart and cause it to potentially stop and the person to die. Well, we worry about that and we try to prevent that. And, you know, when we talk about cholesterol, there's good cholesterol and there's bad cholesterol. And it seems to be, the nomenclature seems to be the opposite of what it should be, honestly. LDL is low density lipoprotein. That's the bad cholesterol, okay? LDL is the bad cholesterol. Versus HDL, high density lipoprotein, is the good cholesterol, all right? So this LDL, this quote, bad cholesterol, it tends to adhere to the artery walls where it can initiate this buildup of these dangerous plaques. And that's why we call it the bad cholesterol. You don't want a high LDL, right? You want a high HDL. That's the good cholesterol. And that's fine because we use that to actually, you know, make our cellular walls and things like that. The membranes, they're part of the membranes, right, of the cell. But the LDL cholesterol is very sticky and it wants to adhere, and that is not good. And so what happens, and just to kind of clarify, is it's not so much, you know, as we take a look at the vasculature with these cholesterol plaques, what you would think is that the cholesterol itself is in the vessel itself. It's actually not. It's in the tissues of the vessel, right? This is why you can't just go in and scrape it out like, you know, a big piece of lard in a tube. You can't just pull it out because it's not actually in the vessel itself. It's not in the lumen of the vessel. It's in the wall of the tissues and pushing into the vessel, as you can see here. And that's why it's so difficult for us to treat for so many years. But now what we figured out is we can put these stents in, right? And this is exactly what they do. This is exactly what the stents look like. So you've got the vasculature here, and they will thread the stent up through one of the major vessels to the region and then expand the stent. So now the mesh is holding the vessel open against the pressure of that plaque that's in the cholesterol plaque that's in the vessel wall. Okay? So that's how these stents work when someone actually has a plaque. But the goal is to not get there and to make sure that we are, you know, really watching our cholesterol, making sure that our LDL is low, our HDL is high, and that we're eating foods that have the good cholesterol, right? And this high-density lipoprotein, the HDL, this good cholesterol, it tends to actually remove cholesterol from arteries and deliver it to the liver cells where it can be broken down. So that is a good thing, and that's why we want, you know, a higher level of this HDL, because it's good stuff. It's cleaning out all the arteries and getting all that bad cholesterol and where it's supposed to go and to get cleaned up by the liver. All right, we are almost there. I want to tell you a little bit about the lymphatic system. It's a totally different system. It runs parallel to the circulatory system. And this is where all the extra fluid in the tissues can move to, okay? The lymphatic system runs close to the circulatory system throughout the body, and it plays a supporting role in the process of circulation. All right, so the lymphatic system basically is all the extra fluid that's in the interstitial space, that space between the cells. It's going to flow into the lymphatic system. And so it's keeping that balance of the pressure, the fluid pressure in the tissues. But also the lymphatic system is sort of the extra highway that the white blood cells and the immune cells can actually travel through. Because remember, they have to surveil every cell and every portion of the body. And so the lymphatic system connects into these lymph nodes. So we've got the lymphatic capillaries. Again, they have valves, right? So things flow in the proper direction. And they end in these lymph nodes. And this is where the lymph nodes are where the white blood cells, a special portion of the white blood cells, actually reside. And so a huge part of the immune system and the lymphatic system is part of that. So the lymphatic system is helping to maintain the fluid flow in the body and also to provide a region for the white blood cells and the immune system to surveil the body. Okay? So, functions of the lymphatic system. Number one, recycling. The lymphatic system recycles fluid and proteins that diffuse from the blood capillaries during circulation, and it sends it back into the bloodstream. It helps in fighting illness because as lymph circulates throughout the body, the white blood cell packed lymph nodes remove dangerous materials, including bacteria, cancer cells, viruses. The lymphatic system helps by retrieving nutrients. So we've got these little projections that extend into the small intestine to absorb lipids from the digestive tract and shuttle them to the bloodstream. So very important, extremely complicated, and we're still learning a lot about the functions of the lymphatic system. But when it goes awry, here you've seen this before in a different person. This is elephantitis. This happens due to filariasis, the little nematodes that actually get into the lymphatic system through the bite of a mosquito, and they damage the valves in the lymphatic system. And because, valves are meant to things flow one way, and lymphatics are basically keeping the fluid in the tissues at the proper state. And when that goes awry, then suddenly we get this huge amount of swelling to the extremities, and this is elephantitis. And as you can imagine, it is extremely uncomfortable. It can be just devastating to a person's life when that happens. All right. We're going to jump into, this is an elephant swimming. If you didn't recognize this image, we're going to jump into gas exchange and how that's happening, how we actually breathe, right? We talked about why we breathe because of cellular respiration, right? That we need the oxygen for the Krebs cycle, really important. We talked about how the oxygen gets to the cell. We just went through the circulatory system. But now let's take a closer look at how the gas is actually getting exchanged between the blood and the cells themselves. All right. So in large multicellular organisms, gas exchange is this two-stage process. So number one, respiratory gases are exchanged between the external environment and the organ's circulatory system. We breathe air in, right? And then these respiratory gases are exchanged between the circulatory system and the cells involved in the cellular respiration. So when we breathe the air in, we have the oxygen that's coming into contact with the red blood cells inside of the lungs. So air outside is going into the lungs, and it's contacting the capillary beds, the red blood cells there, and we have this gas exchange. Now the red blood cells are taking that oxygen to the cells themselves in the capillary bed of the tissues and making that gas exchange. At the same time, the cells are releasing the CO2 from their cellular respiration, getting exchanged back into the bloodstream. So when the blood returns to the lungs, that CO2 is getting exchanged as well. So it's a two-way exchange that's actually happening. All right? Now, this can happen in different ways in different organisms, right? So Diversity in gas exchange system. So direct diffusion. Gas exchange occurs directly between cells and the environment. So this occurs in single cell organisms and small organisms with low metabolic demands. So they're actually exchanging right here on their tissues. Right here, here's the cells, here's the external environment with the oxygen being diffused into the cells and the CO2 being released out to the environment. There are no lungs. This is direct diffusion, okay? Or in some organisms, like the starfish, you have these protruding respiratory sacs. So these balloon-like sacs that increase the surface area for gas exchange. And this occurs in sea stars and other echinoderms with low metabolic demands as well. So here's the respiratory sac, and this is where the gas exchange is happening. No lungs. And then when we get to fish, we have gills. And basically, gills are these elaborate extensions of the body that exchange significant amounts of gases dissolved in water. And so fish and many marine vertebrates, such as lobsters and clams, also use this method of gas exchange, these gills, right? When we have insects, they have something called tracheae. And tracheae are these network of branching tubes that are connected to tiny openings on their body called spiracles, as you can see here. And this is where the gas exchange is happening for them. Again, no lungs. But when we get to mammals and most land vertebrates, we have lungs. All right. So here is what's happening. And lungs are these internal organs that we have with highly branched, moist surfaces. And this is where the gas exchange is actually happening, right here in the alveoli of the lungs. So we have the air from the outside coming in here, if you follow my cursor, going into the lungs, and it's just a constant branching system. So we have the main bronchi splitting off into two left and right bronchi, going to the left lung and the right lung, the sides of the lung here, right? And continuing to branch down smaller and smaller until we finally get to this end point, which is called the alveoli. And this is where the gas exchange is actually happening. Here we have the capillary bed. And so the oxygen is diffusing directly to the red blood cells here in the capillary bed at the end of the alveoli. And the CO2 is coming out of the capillary bed into the alveoli in order to release it. And so this is all happening because of this molecule called hemoglobin. So each molecule of hemoglobin is this tangled mass of 4 polypeptide chains with 4 molecules of iron that create 4 seats to which oxygen can attach. So you can see here we've got 4 places on a hemoglobin molecule that oxygen can attach, okay? And this is why we need iron for our red blood cells, right? And you get iron deficiency anemia. So this is why iron's really important, one of the reasons. So the hemoglobin molecule is like an oxygen shuttle bus that picks up oxygen in the lungs and transports it to the tissue. So it is very attractive to oxygen. So depending on how many open seats it actually has. It has 4 in total that it can fill, okay? Really important to understand that. But it turns out that when we breathe, we are actually breathing because of the partial pressure of oxygen. What does that mean? So here we have a person and they're at a certain elevation. And here is a person at a higher elevation. Now, there's something called air pressure. Air pressure is the force of all the particles in the air pressing against you. So when we breathe, the air that we breathe is not 100% pure oxygen. It has other molecules that are in it too. And so the air pressure is the force of all the particles in the air pressing against you. And this force actually decreases as elevation increases. The higher you go, the less force of air pressure there is because there are fewer particles pressing against you at a higher elevation. Okay? Stay with me on this. Now, so what is the partial pressure of oxygen? I told you we breathe, we're able to take in air because of the partial pressure of oxygen. So the partial pressure of oxygen is the force of oxygen in the air pressing against you. So here we've got all the atoms that are in the air pressure that are pressing against you at the elevation that you are at. And oxygen is one part of that, right? That is the pressure of oxygen. Now, the partial pressure of oxygen is usually about 21% of the total air pressure. The air that we're breathing in, the air pressure, is only about 21%, all right? Now, when hemoglobin encounters a high partial pressure of oxygen, such as inhaled in the lungs, then hemoglobin gets packed with oxygen. And so here we can see hemoglobin binding oxygen. But when there is a lower partial pressure of oxygen, so hemoglobin, look at the oxygen molecules here, okay? So when hemoglobin gets to the capillaries and there's less oxygen there, then when hemoglobin encounters a low partial pressure of oxygen, such as in the capillary bed of an active muscle tissue, then hemoglobin releases the oxygen. So what hemoglobin is doing is actually saying, listen, where I am, is there more oxygen pressure out there? If there's more oxygen out there, then I'm going to take oxygen in. If I get to a place and there is less oxygen out there, I'm going to give up oxygen, okay? Now that's really important to understand, that hemoglobin is working on this oxygen partial pressure. It's actually, sensing the amount of oxygen outside of where it is, in order to determine if it's going to take oxygen in or if it's going to deliver oxygen. Now, there's this binding curve for hemoglobin. So here we have the oxygen saturation for hemoglobin. Here we have the partial pressure of oxygen. Now, oxygen is delivered during normal metabolism. So the oxygen saturation is high. The hemoglobin gives up only one oxygen molecule in the body tissues before returning to the lungs. Okay? That's right here. See our 4 spaces? The hemoglobin spaces for oxygen, right? But, excuse me, oxygen is delivered during high physical exertion because the oxygen saturation is lower. So hemoglobin dips into its reserves, releasing 2, 3, or even all 4 of its oxygen molecules before returning to the lungs, right? So depending on where this oxygen saturation is, determines how much hemoglobin how much oxygen the hemoglobin is actually going to release, okay? And so there is a point where too much oxygen is actually not good. So you would think a lot of people go to these oxygen bars. I want to just breathe some oxygen all the time, get lots of oxygen. Well, it's not really good, right? Because we're not meant to breathe 100% oxygen all the time. We're meant to breathe at about 21%, okay? And what can happen is you can actually get this delirium that can happen. You can get this oxidative damage that can happen if you're in a constant hyperoxic state for too long. And so we are very, very aware of that when a body is under anesthesia. And you're hooked up to oxygen, and yes, we want to make sure you have enough oxygen, but we have to be careful that we're not that we're watching the oxygen saturation of the blood and measuring that actually gets measured with really long surgeries, so that that we can make sure that the tissues are being properly oxygenated and that we're not getting into this hyperoxic state, which can be damaging. All right. So, what do you want to remember? There's an oxygen binding curve for hemoglobin. That oxygen is delivered during normal metabolism. Um, Oxygen saturation can be high, and when that happens, hemoglobin will only give up one oxygen molecule in the body tissues before returning to the lungs. If the tissue's already pretty much oxygenated, you're sitting here, you're not really doing anything, you're not really moving. So you're not having a whole lot of exchange, right? Because when the hemoglobin gets to your tissues, it's just like there's not a lot of metabolism going on. There's a lot of oxygen that's in this region already. I'm going to give up one and then, move along, right? However, when there's a high physical demand, then again, looking at this oxygen curve for hemoglobin, then the oxygen saturation in the region of the tissues is low. And then hemoglobin is going to dip into its reserves and just start throwing out, releasing 2, 3, even all 4 of its oxygen molecules before it returns to the lungs to pick up more. Okay? So, also, let's give a little talk about fetal hemoglobin. So, there is a difference in hemoglobin in a fetus. The hemoglobin is more attracted to oxygen, slightly, quote, stickier for oxygen than the mother's hemoglobin, which is why the exchange happens. So, number one, let's just follow it. Follow the flow. You have deoxygenated fetal blood, which flows in close proximity to the mother's blood. It is not admixed. There is not a mixing of the blood happening. Then the fetal hemoglobin, which is slightly, quote, stickier and more attractive to oxygen than the mother's hemoglobin, binds to oxygen as the mother's hemoglobin releases it as they go, the vessels are going by. This diffusion is happening, right? There's no admixing. And so step 3, oxygenated fetal blood flows back to the fetal tissues, delivering the oxygen, and then the circuit begins again. And so this is an enlarged area of what's happening in that region. So there's a different type of hemoglobin that fetuses have. All right. We're going to take a quick look at the gas exchange in fish, which we saw before with the gills. You can take a look at this sort of on your own. Here we have the gill structure, we've got the gill arch, we have the filaments, and then we have the laminae, and then we have basically the capillary beds where the blood exchange is happening. Okay, so this is sort of this huge overview of the human respiratory system. So we can see that the air comes in through the nose and the mouth. We have this common area in the back called the pharynx. And then we have the larynx. This is where the vocal cords are. This is how we actually speak. And then the air comes down the trachea, goes into the bronchi, and this is the major bronchi branching into the left and the right bronchi, as we can see here, going to the left and the right lung fields. And so in terrestrial animals, the respiratory system provides the route for inhaled air to meet the blood vessels of the body and to acquire the oxygen necessary for cellular respiration and to remove the carbon dioxide, the waste byproduct of cellular functions. So as we take a closer look at the gas exchange regions of the lung, here we can see an image that's colorized of the alveoli. Here we've got a major bronchiole coming down here, the alveoli here. And they look like just this very lacy pattern. And here we have the blood vessel from the capillary. And what's happening as we look at this illustration, you can get a closer look. Here we have, if you follow my cursor, the bronchi coming down. We have the vasculature coming from the heart and also returning to the heart. So remember, coming from the heart, it's going to be deoxygenated, right? Because it's coming from the heart, which came from the body, deoxygenated. And so each alveoli which is the very, very end, as these bronchioles just, dilate and branch and get smaller and smaller. The very end are the alveoli. And this is sort of the cross section of what's happening in the alveoli. This is where the gas exchange is actually happening. And so you've got the capillary bed that's encircling each alveoli. And so the carbon dioxide diffuses from the blood into the alveoli, out of the body as exhalation occurs. And then inhaled oxygen diffuses from the alveoli into the blood based upon that oxygen concentration gradient, right? And it turns out there's about 300 million alveoli in each lung with a total surface area the size of an entire movie screen, which is incredible. But there are different respiratory adaptations that have evolved that can enable birds to do gas exchange with great efficiency, even at high altitudes where oxygen availability is really low. So here we have these whooper swans that you see in the image here, which have been recorded migrating at altitudes of up to 8,200 meters, where the ambient temperature is negative 40 degrees centigrade. So how are they still doing gas exchange? Well, it turns out just like scuba divers that have an extra tank of oxygen on their back, birds have the same thing. They have these air sacs. And these air sacs basically act as an extra tank of air, oxygen, for the bird. So we have the trachea coming down. Here we can see a branch with an air sac here going to the lungs. These are the anterior air sacs, which are shown here in the illustration of the bird. And then we have the posterior air sacs, which are here. And this is a very simplified version of their respiratory system, but basically explaining how they're able to still fly and exchange gases at such very high altitudes at such low oxygen pressures. It's because they have these air sacs. But it also turns out that these air sacs tend to be sources of infection. In birds, we can get, in the veterinary clinic, a lot of infections that we see in just household birds coming in where their air sacs get infected. But that's how they're able to actually fly at high altitudes. And so here, let's just take a closer look here. So we've got with an inhalation, we've got the fresh oxygen-rich air moving down the trachea, inflating the posterior, quote, waiting room sacs, as well as the lungs. And at the same time, the oxygen-poor air is expelled from the lungs, inflating the anterior air sacs. And then when the bird exhales, excuse me, the posterior air sacs deflate, pushing the oxygen-rich air into the lungs. So at exhalation, the posterior sacs deflate, pushing oxygen-rich air into the lungs, and then the anterior air sacs deflate, pushing oxygen-poor air out of the trachea. So it's a little complicated. I don't need you to memorize this, but I just do need you to know that birds have these air sacs, and this is what enables them to continue to fly and exchange oxygen at extremely high elevations. And so in birds, unlike in humans, air is moving in one direction through the lungs, and during both inhalation and exhalation, fresh air continues flowing throughout the lungs, okay? But when we look at the mechanics of breathing, so breathing is made possible by the following structures in the chest cavity, and this is now looking at the human. We have the trachea coming down, branching into the bronchi. We can see the lung fields here. They are covered by ribs for protection. We have intercostal muscles, between costa, the ribs, muscles. So these are muscles that are between the ribs. the intercostal muscles, and then we have the diaphragm. And this is how breathing is actually made possible. So the mechanics of breathing. In reptiles, birds, and mammals, breathing occurs in 2 steps. So I want you to pay particular attention to the size of the lungs outlined by the dashed lines during inhalation and exhalation. So during inhalation, the diaphragm and the intercostal muscles contract. The diaphragm is pulled lower and the rib cage expands as air is sucked into the lungs. So it's this pulling down of the diaphragm. It's actually sucking air into the lungs. And then during exhalation, the diaphragm and the intercostal muscles relax and the chest cavity returns to its original size and air is forced back out to the trachea. I do want to show you a movie of this because I think seeing it in motion actually helps to understand what's happening. So if you look here on the screen, you will see our person that is actively breathing in 3D. We can see the vasculature that's coming down from the head and neck. We can see the trachea here. But what I want you to watch is the movement of the diaphragm and the expansion of the chest. You can see the heart right back here. So as the diaphragm contracts and pulls down, you can see the chest expanding. It relaxes, air goes out. Contract, chest expands, relax, air goes out. Contract, chest expands, relax, air goes out. And so this is the dynamics of breathing, and actually a really clear look at the respiratory system in action with everything in its proper place. We can see the lung fields here, we can see the heart that is here, we can see the diaphragm, we can see the descending aorta and also the inferior vena cava right here. So you can see the major vasculature as well. And so this is the dynamics of respiration. Now let's talk about breathing at high altitudes, because as you know, if you were to try to climb Mount Everest, for example, you have to take oxygen tanks with you because we as humans cannot breathe at that height. We're going to see why. So there are different organisms that can. So llamas can actually thrive in high altitude, low oxygen environments because they produce a stickier form of hemoglobin that has a higher affinity for oxygen. So that stickier basically is just another way of saying, common term, of saying a higher affinity for oxygen. So they have a different type of hemoglobin. Whereas for us, we actually have to slowly start to train at higher elevations for a couple of reasons. Number one, we're going to create more mitochondria at higher elevations, right? So that we're able to do more metabolism, more cellular respiration. So we will actually generate in our cells more mitochondria. We will actually create more red blood cells in response to being at a higher elevation. Depending on the person, this can take a week to 2 weeks for those changes to actually happen. And so you'll see quite a few athletes who are trying to increase their performance level naturally without drugs and that sort of thing by training at higher elevations, because you're going to get more red blood cells, you're going to have more hemoglobin, you're going to have more mitochondria. And so when you come down to compete at a lower level, you've got all of this extra metabolic and respiratory capability. And so a lot of training actually happens at higher elevations. But also, before you even can scale up to, say, a Mount Everest, you can't just go straight up to the top, right? Even if you have the oxygen with you, you have to begin to train to get to these higher levels, because your body has to begin to compensate and you have to give it that time. And so what's happening is You're getting this additional red blood cell production. You're getting this increase in blood and capillary volume, this increased number of mitochondria. You're getting a increased DPG, which is diphosphoglyceric acid in your red blood cells, which is going to reduce the hemoglobin stickiness and cause it to release more of the oxygen that it carries. Remember, I told you that when it gets to the tissues, it's kind of looking at that oxygen concentration gradient, and it may only release one molecule of oxygen. Well, we want it to release more to the tissues, and so training at higher elevations will actually increase this DPG so that it will have a greater propensity to do that. All right, so the reason we can't breathe at higher elevations is because remember that partial pressure of oxygen. Remember that slide that I showed you where it shows the oxygen that is in the amount of air, and the fact that it goes down the higher you are in elevation. And remember that, if we go up in elevation, and here, let me get to that slide for you. There you go. If we go up in elevation and we have less oxygen here, and our hemoglobin is reading basically the amount of oxygen that's in the alveoli, and if there is not a lot there, it's not going to take up any oxygen, right? It can't. It's not, because it's actually reading the percentage of oxygen. So when you get to a much higher elevation, it's not taking in the oxygen. The hemoglobin is not that attracted to the oxygen. And so there may be, oxygen in the air, but the hemoglobin's not actually going to take it in. And that's why at a certain elevation, humans cannot exist, because we will not exchange the oxygen. So that's really important to remember, that there are limits to where humans can go on the planet, secondary to the way we actually breathe, which is based on the oxygen concentration and our hemoglobin type and its propensity to have an affinity for oxygen, right? But we can train to increase that higher level. We can't just immediately jump to those higher elevations. We will feel sick. Some people can even die. And we have to slowly sort of work our way up to the limit of where humans can go. All right, so I do want to break out and show you some x-rays. Okay, so I wanted to give you a look at what's actually being seen on an x-ray when you go in for an x-ray of your chest. And so this is a really nice look at it. I put the link into the supplementary materials in your lecture, so you can go through this for yourself and have fun and kind of see where the structures are and what's being seen. So here are the clavicles. These are the collarbones. You can see them showing up here. And when you think about x-rays, basically just, we already talked about the x-ray spectrum and what it is. And essentially what you're looking at is, it's almost like you're holding up the body to a very strong light. And you're looking at the shadows of everything coming through. And so the more density you have to a structure, the lighter it's going to be. And that's why bones show up in this white form, right? And air is dark. So here we have the lung fields here, and here we have the bones. So let's go through the bones. So we've got the clavicles right there, which are the collarbones. Here we've got what's called the manubrium of the sternum. Here we've got the scapula. These are your shoulder blades showing up here in green. And we've got the spinous process of the vertebrae that you can see here, which continues all the way down. They just highlighted these here. We've got the body of the vertebrae of the spine that you can see here on the x-ray. This happens to be a female. We can see the breast shadows here. That's what those regions are. That's the density of tissue. This is showing you the cardiac width. This is the heart. Right here, it goes from here all the way to here. Here we can see what's called a gastric bubble. This is basically just air in the stomach. This is the stomach sitting right here on the left side of the person. And here it's just showing you the full thoracic cavity, the thoracic width. Okay. And so basically that's what you're looking at on an x-ray. And so when you're looking at the x-ray of a chest, you're looking at the lung fields. You want to make sure, because this is the diaphragm here, if you follow my cursor, you want to make sure that the lung fields are nice and dark, which means that the person is moving air. You can see some of the vasculature in the lungs here as this little branching density. Here we can see the ribs that are arching out, coming off the vertebrae. But you want to make sure there's no like what looks like fluffy clouds, which would be indicative of like say a pneumonia or solid type of circular masses, which would be tumors in the lungs. Sometimes you can have foreign bodies where, you know, an individual has breathed in something and it's gone into one of the lung fields, and you'll see what appears to be sort of these white fluffy areas of inflammation that can be there. But mostly what we tend to find is pneumonia and tumors, that sort of thing. Certainly we're looking at the heart to see whether or not it's enlarged. So there's a lot that you can see on the x-ray. And one of the things that we do when we look at an x-ray is, you know, you want to look at the main things that sort of jump out at you. Of course, the lung fields, because you took an x-ray of the chest. Obviously, you wanted to look at the lungs and the chest itself. But you also want to take a look at everything that you can see in the x-ray. You want to look at all the bones, all the structures that were actually picked up in the x-ray to see if there's any abnormality that is present that you didn't even know existed, even though you were doing an x-ray to look for something else. So when we look at an x-ray, we look at the totality of it. And this is the chest x-ray. And when you go to this site, I'm going to see if I can click through. It's going to give you all these other x-rays that you can look at on your own. I just, you know, went to the thorax, and I went to bones and landmarks. There's plenty there. There's the head, x-rays of the head, x-rays of the abdomen, shoulder, leg, pelvis, and it's giving you all the bony structures, the neck, the back, the arms. So have fun with that. And the last thing that you're going to be doing is this week is doing your lab. And so make sure when you go into your lab, what you're going to be uploading for me is this section right here where it says quiz. Once you finish the lab, all the quizzes in the lab that you took while you were in the lab are going to be in this quiz section. And you're just going to print it out. This one's empty because it hasn't been done, but you'll have all the quizzes right here. You're just going to do a screenshot of this full quiz page, and with all your quizzes and responses in there for me, and that's what you're going to upload. Here, make sure you take a look at the notebook, because as you go through the lab, this is going to be filling things out for you, detailing the steps. There's a reference area so you can look things up. And of course, where your patient information is. There's a save and resume. Remember, if you close your browser, it may not remember, you know, where you were. So just kind of be aware of that if you do need to stop, that it may or may not save it. So just kind of be careful with that. It's not a super long lab. It's actually pretty interesting. You're going to be diagnosing patients, and so it's going to tell you all about it. You've done these HHMI labs before. I actually think that they're very useful for extremely detailed information to walk you through. So you're going to be doing cardiac ultrasound. You're going to be doing auscultation of the chest. You're going to be diagnosing a patient, and you're also going to be integrating your knowledge of genomics in a portion of the section to try to get to the diagnosis. So I hope you have fun with that. All right, I've got a couple of other supplementary materials that I'm going to be putting up in the lecture for you. If I can get them to record just fine. One is a really good workshop that was done at this last conference that was all about cardiac arrhythmias, what to do when you see them. So this is real world, and what happens in the clinic when you see the cardiac arrhythmias that we talked about with the ECG, what they look like, and the drugs and the medications, the treatments, what we do when we see these things. So feel free to take a look at that if I can get it to go ahead and upload okay. And the other one that I'm going to try to upload for you, that I'm going to try to play at the end of this lecture. So after I say goodbye, what's going to come in, hopefully, is going to be the video on cardiac arrest. So this is a patient that is in the hospital, and it's a training video, but you're going to see what actually happens during a cardiac arrest. You're going to see the cardiac arrest team at work going in to save this patient's life from the point of realization that the heart has stopped to getting it started again. They're going to go through both an open and external CPR. They're going to do an open heart massage. They're going to put the paddles on to restart the heart. And it's actually pretty interesting to get to see what really goes on in the real world during a cardiac arrest. So hopefully I will be able to attach that to the end of this video. And if you have any questions, please let me know in the Zoom. And if I can't get it to attach to this, the video at the end of this one, if it's not there, that means that I will probably be showing it in the live Zoom sometime this week. So if you jump into the live Zoom, I can go ahead and get that playing if I can't attach it to this one. All right, see you guys next time.